Okay, this is a little kind of a short lecture on cell communication, how cells communicate. This process was actually a man here in Dallas at Southwest Med School got a Nobel Prize for G proteins, and that opened up a whole new door of understanding for how cells communicate. I don't think he's here anymore because those people usually get taken away to some other research institute. So, cell communication, how cells communicate. Let's look at this chapter. It says cell signaling. Okay, regulation and control is essential at all levels of biology. It is. Remember, I've told you before that the cell is very orderly, very organized, and it uses energy when it needs to, and when it doesn't, it turns things off. And so when processes are needed, they're carried out, and when they're not, they're turned off. Now, some cells can regulate the activity of other cells by sending messages, which we'll look at in this chapter also. And these messages, like number 2 here, chemicals from the environment and other cells are the signaling agents. The signaling agents are the messenger molecules that are coming from those other cells. They exert their effect through signal transduction or a transduction pathway. We're going to see that once these messenger molecules or signal molecules have bound to a protein in the membrane, that triggers a series of events. One of them is it binds, you have a transduction pathway where like molecule A to molecule B is converted to molecule C, converted to molecule D, and you get a cellular response. So this chapter really deals with 3 things. Binding of a messenger molecule or a signal molecule to the membrane receptor, a transduction pathway, a series of biological pathway, and then the end result, which is called a cellular response. So here's just an example here. It says chemical signaling, how cells communicate with each other is what that means. And the little example has to do with some yeast. It says coordination of yeast mating behavior, how yeast find each other. Now, the yeast is called A type, produces an A factor that it releases to the environment. Well, there's a second one, alpha type. Alpha type produces an alpha factor into the environment. The binding of factors to membrane receptors of the opposite types causes them to move together and to fuse. So when A type produces A factor, they're binding sites for A factor on the alpha cell, and it will home in on the source of the A factor. Now, when the alpha cell produces alpha factor, there are binding sites on the A type for alpha factor, and it will also seek out the source of alpha factor. So they're drawn to each other, seeking out the source of the factors. A signal transduction pathway, steps by which the signal, the message, is converted to a specific cellular response. Remember, I told you that there are 3 parts to this. There's a binding of the messenger molecule to the receptor, a transduction pathway, and a cellular response. Here are these 2 cells. Look at the one on the left. This is the A type yeast cell. And you see that, look at the bottom left, a little arrow down here is pointing to a little golden color sphere. That's going to be the A factor. Well, look over here on the alpha cell, and you'll see that the A factor has binding sites. So they're receptors for A factor. You see several spots, even on the bottom right, it says receptor for A factor, and you see it's going to bind there. Well, look at the alpha cell, and you see that the alpha factor is a little green triangle. And you see going across the top from right to left, the A factor is finding receptors for, the alpha factor for alpha factor on the A cell. So each of these 2 cells, the A cell and the alpha cell, are going to be homing in on the source of those factors. You see that they find each other, they bind together, fuse their nuclei, and their DNA is mixed, and then they will bud off and form new cells, which are hybrids of those 2 cells. Types of communicating. I've got 3 of them here. Local regulators. It says work between cells located near each other. That's true. Paracrine, you have paracrine factors, which will only affect the cells in the general area. Now, you've probably noticed sometimes you'll have a little spot on your arm or your leg or something, and it's red, and then the cells around it are kind of red, but that's the only place that's really bothered. That's paracrine factors. Something is affecting your skin, and the cells around there are sending information to the surrounding cells to handle that problem, whatever it is, a bacterium or some molecule that's causing an irritation, but it's only in that area. So that's local, not systemic, it's local. Now, synaptic signaling, we haven't talked about synapses, but when a neuron sends an impulse to another neuron, they don't actually touch each other. There's a space in between them that has a little gel there, and chemicals are sent across that gel from one neuron to the next neuron. That's called synaptic signaling. And you look at this definition up here, work between cells located near each other. So it's a one-to-one signaling. Neuron A sends the impulse to neuron B. B sends one to C. C to 1D. That's local. Now, distant regulators, says, impact cells located some distance from each other. Well, it can be local or distant in this case for hormones. Like it says, hormones may be local or distant. Now, let's look at a distant. Insulin secreted by your pancreas. That affects all the cells of your body. So that's distant. Thyroxine, which is secreted by your thyroid gland, that controls the metabolism of all your cells. That's local and distant. So both of these are local and distant. There are some that are more specific, like prolactin, that's released by your pituitary, only affects the cells of the breast for making milk. So that's going to be pretty specific. That's going to be a distant only, not local. So hormones can be local or distant, and there are some that are just distant. Cell to cell, this is how cells communicate through their cytoplasm. The gap junctions in animal cells, cytoplasms communicate with each other, and so the cells stay in homeostasis. In plasmodesmata in plant cells, they have protein channels that allow their cytoplasms to communicate also, and so the cells of the plant remain in homeostasis. They know what's going on in each of the cells. That's how they communicate, cell to cell. Here are some images showing local signaling. Here's the paracrine, where this cell is stimulating just the cells around it. That's it. So whatever it's secreting is only affecting the cells around it, like that little red spot on your skin. That's the only place that's being affected. Synaptic signaling, this is a drawing of a neuron here. And you see that it is communicating with the next neuron, but there's a synapse there, a space. And it has to chemically transfer that information, that impulse across a cleft. So it's one to one. So that's a local signaling, synaptic signaling. Hormonal signaling, that goes into your bloodstream, and so it goes systemic. And so the cells that make the hormone send it directly into your bloodstream, and it goes throughout your body to the target cell. The target cells could be distant or they could be local. If they're local, the hormone's still going throughout the body, but it's only affecting those local cells, the cells it's meant to affect, which are called the target cells. Here we're just showing some gap junctions in plasmodesmata, how in gap junctions in animal cells, the cytoplasms can communicate with each other, and so the cells remain in contact with each other, and they know what's going on. You know, like a virus infection, this one could signal this one there's a virus in here, and this one starts making the interferon to prevent the virus from replicating. Here's plant cells here, and they also have protein channels called plasmodesmata that allow the cytoplasms of the plant cells to communicate with each other. This is one that's an interaction that says cell surface molecules, cell to cell. When this cell butts up against or binds to this cell, there's a receptor here, and it causes the cell's activity to change. Three stages of cell signaling. That's what I was telling you just a minute ago. This whole chapter deals with this. Cells must have the proper signal receptor system before a signal can elicit a response. Wow. Cells must have the proper receptor system, it's a protein, before a messenger molecule can cause a response by that cell. So it has to have the receptor for the messenger or the signal molecule to bind to before it's going to have an effect on that cell. Remember, all hormones are not meant for all cells of the body. They have their target cells. And so not all of the cells of the body have those protein receptors in their membranes, only the ones that hormone's meant to affect. And other molecules for changing the activity of cells. Again, they're made, the messenger molecule or the signal molecule is made for a specific cell. So it says 3 elements of the signaling system in the target cell. Okay, for the target cell is the one that the messenger molecule or the signal molecule is meant to affect, is that type of a cell. Signal recognition, there is a way that messenger molecule can bind to a protein, so it was made to bind to a specific protein for that target cell. So it binds. Signal transduction, once the messenger molecule binds and is recognized and it binds, it causes a pathway to occur inside the cell on the cytoplasmic side. It's called the signal transduction pathway. So it causes possibly an enzyme to be turned on, which converts molecule A to B, and then B can be converted to C, and then C to D, D to E, E to F, until you get the cellular response. What it's supposed to do, produce something or stop producing something. An example, epinephrine and glycogen metabolism. Epinephrine is adrenaline, if you know the other word, and glycogen is a polymer of glucose. Now it says epinephrine breaks down glycogen. Well, that's true in a way. So epinephrine is going to bind to a receptor. See, it requires intact cells, plasma membrane receptor for epinephrine to bind to. When it binds, it activates intermediate steps inside the cell. That's the transduction pathway, signal transduction, which activates glycogen phosphorylase. ASE is an enzyme ending, and that's what's going to break down the glycogen. So the epinephrine starts the process to activate this enzyme to break down glycogen. Here's a good little set of images here for showing this. Here's your messenger molecule over here on the left. You see that it's binding to a receptor. It's in this membrane. This gray structure is the cell membrane. Here is plasma membrane. And you see that it fits perfectly. So this signal molecule, this messenger molecule, was made to bind to that receptor in that target cell. When it binds, we're going to see in just a minute on the next slide or so, this causes this protein to change shape. That triggers the transduction pathway, the starting of a chemical pathway, converting molecule A to B to C to D, and you get a cellular response. So reception of the messenger molecule, transduction pathway, and cellular response. This is how cells communicate. And these 3 processes are carried on every time one cell communicates with another cell. So, signal reception and initiation of the transduction pathway. Okay, so we have several words for the messenger molecule. Chemical signal, that's a messenger molecule, or a ligand, same thing, binds to the receptor in the membrane, the protein receptor. Most receptors are plasma membrane proteins. Okay, just accept them as being membrane proteins. Those are the receptors. When the messenger molecule binds to the membrane proteins, it causes an alteration of the receptor's conformation. It makes it change shape. So it changes shape. When the messenger molecule binds to that membrane protein, it changes shape. The altered receptor interacts with cellular molecules on the inside. That's going to be the cytoplasmic side of the membrane. That'll start your transduction pathway. Now, here's the gene, the families here. The first one is G proteins. This is the one that that man about, 20 years ago got a Nobel Prize for. Single polypeptide. Okay, it's one polypeptide, one protein. Threads through the membrane 7 times, and we'll see on the next picture how that little image is drawn for threading through the membrane, but it's a membrane protein. Interacts with G proteins, which are located on the cytoplasmic side of the membrane. So this polypeptide here is an integral protein. It goes all the way through. And when it reshapes, when a messenger binds to it and it reshapes, that's going to interact with the G proteins on the cytoplasmic side of the membrane. The reconfirmation of that membrane protein interacts with the G proteins on the cytoplasmic side. Now, this has the G stands for guanine, so the binding of GTP is going to activate that G protein. The binding of GDP inactivates that G protein. Well, we're going to see that this G protein has GTP ACE activity. Once G protein's bound to it and is activated, it begins to break down the G protein to GDP and inactivate itself. So let's look at a little picture here. In just a minute. Activated G protein interacts with another protein, often an enzyme, thus activating the enzyme activity. A lot of times it's going to have to do with phosphorylation. The activation of the G protein is temporary. So once the messenger binds to the membrane receptor and the G protein is activated by a GTP being bind to it, it's only temporary. The activated G protein has GTP ACE activity, which hydrolyzes GTP to GDP and turns itself back off. G proteins are involved in the regulation of many critical metabolic and developmental processes. That's why this was so important, because it opened up a whole new world of scientific study when G proteins were explained how they work. Now here's this protein. See, we draw them like this on the pictures, and there's that little receptor site there, which you can see is the receptor site right there. And it said bind goes through 7 times. So there's 1 in, that's 2, 3, 4, 5, 6, 7. Transmembrane portions there. Here's the binding site for the messenger molecule out here. And here's on the inside, the reconfirmation part, that segment that interacts with the G proteins. So this kind of gives you an idea. On your picture, it's just going to show something like this. But blown up, it kind of gives you a little bit more detail about the parts that are going to interact. So here is, like it says here, inactive form. Here is the membrane, the protein in the membrane. This has the receptor on it. Here's your inactive G protein. See, there's your GDP on it, so it's inactive. Here's an enzyme that has to be activated. Right now it's inactive. So let's look at the second picture. A signal molecule, a messenger molecule, binds to the membrane receptor, which causes it to reconform, which results in the joining of GTP to this inactive G protein. When it's activated, it goes over and activates this enzyme, and then it's going to be deactivated. This enzyme is the first enzyme in a pathway. There's your transduction pathway, all these arrows, to a cellular response. Now that happens once, because now this enzyme loses its phosphate because it was phosphorylated by GTP. The G protein is inactive, and this messenger molecule has been broken down. So this happens once. One binding, one transduction pathway, one cellular response. And then another messenger molecule has to bind for it to happen a second time. So as long as these messenger molecules are being produced, they're binding to the membrane receptors and keeping those cells doing whatever the cellular response is to turn on a process or turn off a process. Now, tyrosine kinase, this is the second family here. This one's kind of unusual. See, it says external receptor, yes, and an internal domain with tyrosine kinase enzyme activity. A kinase is an enzyme that breaks down ATP. Remember, kinetic for energy. So kinases break down ATP, and they're going to phosphorylate tyrosine. So this is a pretty cool looking receptor here. There are two parts to it. There are two receptor units that join together, forming a dimer, a receptor dimer, two pieces, two units. Dimer formation, when they come together, so the messenger molecule is going to bind to the receptor sites. The two dimers come together, so dimer formation, and when they come together, it activates tyrosine kinase on the cytoplasmic side, and that kinase is going to phosphorylate the tyrosine molecules, and we'll see what that looks like in just a minute. So it says transfer of phosphate from ATP to tyrosine amino acids on the cytoplasmic domain of the receptor. So when the dimer comes together, tyrosine kinase is activated, and the tyrosines on the cytoplasmic side are phosphorylated. Phosphorylation activates a relay protein. So there were some proteins that were not, like enzymes, that were not active. Well, now that the tyrosine is phosphorylated, it activates the relay protein, which starts the transduction pathway, and you get a cellular response. And then it all comes back apart. Remember, it only happens once per binding. Many different proteins may be activated, depending on which one it is. Dephosphorylation of tyrosine kinase receptor results in the termination of the signal process. Because remember, it's only going to happen once. Every binding, it happens once, and then it's deactivated. So let's look at that one. Here are the two parts of the tyrosine kinase system here. And this is the dimer here, two units. Here are the receptor sites, the little cups. And here are the tyrosines on the inside. Here's your inactive proteins. Well, the messenger molecules have to bind to these two receptor sites. And when they do that, the two dimers come together. Okay, so see, there's your signal molecules, your messengers are bound. The two dimers are together. Tyrosine kinase is activated once they come together. See, activated tyrosine kinase causes phosphorylation of all these tyrosines. Their phosphorylation is going to activate these proteins, these enzymes, which are the first step in the transduction pathways. There's two of them here, and you end up getting a cellular response. Now again, that only happens once. It returns back to the normal until more messenger molecules bind, and it happens again. So as long as your messenger molecules are being produced, this will keep happening over and over and over, because it's one binding and then they're broken down. And another set of signal molecules has to bind again for it to happen again. So it's one binding, one transduction pathway, one cellular response every time it works. And then we have the third family, which are ion channels. Now the ion channels, we had protein channels, right? So signal protein binds to the gated ion channel. So there's a receptor site on those channels for a messenger molecule to bind to. And you remember I told you, when the messenger molecule or the ligand or the signal molecule binds, it causes that protein to reconform, to change shape. In the case of ion channels, it causes them to open. So ion channel proteins are for specific ions. Remember, they're very selective. A sodium ion channel only lets sodium go through. Calcium ion channel only lets calcium go through. A potassium ion channel only lets potassiums go through. So they're very specific. Now, on these, when we get to something else like receptors, may be intracellular. So you have some receptors that are actually inside the cell. They're not on these channels or the tyrosines or the membrane receptors there. But some of them can be intracellular. They can be also inside. Hormones like progesterone, testosterone, estradiol, and thyroid hormones, they have intracellular receptors. So they are lipid soluble. They go through the membrane. So there's not a membrane receptor. They're gonna go on the inside to find their receptors. So let's look at the ion channel first. Here's our ion channel. Here's your signal molecule, your messenger molecule binding. And when it binds, the channel opens, allows whatever these ions are on the outside to come on the inside, and their presence on the inside triggers a cellular response It's got a real short transduction pathway for a cellular response, and then it closes again. So one binding, one channel opening, one transduction pathway, one cellular response, and it closes again. Now these signal transduction pathways here, it says the signal, the messenger, the message of that messenger molecule, not the messenger molecule itself, just the messenger, the message is relayed through a series of steps from receptor to the target molecule. So the signal molecule, remember, it just binds on the protein and then it's broken down. So it's one binding, the protein reconforms, and then it reforms back, and that molecule is broken down. So it only happens once. Protein phosphorylation is a common method of signal transduction. So usually, a lot of times when the messenger molecule binds with the receptor in the membrane, it causes reconformation, which causes phosphorylation to start of an enzyme, let's say, to start the transduction pathway for the cellular response. Cytoplasmic protein kinases, remember, kinase is going to break down ATP, assists, helps the transfer of a phosphate from an ATP, because they're going to break it down, to other proteins, some of which may be enzymes. That's a common theme there. So the kinases are going to break down ATP and phosphorylate another molecule like an enzyme. The phosphorylation may serve to activate an enzyme or to inactivate it. Yeah, they can turn a process on or turn a process off. The signal transduction pathway is turned off when the signal is no longer present, when no more bindings occur, no more reconformations of the membrane protein occur, and no more transduction pathways occur, and so no more cellular responses are going to occur. Protein phosphatases can reverse the effects of protein kinases. So where a kinase will break down an ATP to phosphorylate a molecule, phosphatases can put that ATP back on, or pull it off the molecule and put it back on ATP. Here is something called a phosphorylation cascade. Here's a signal molecule, a messenger, bound to the receptor. The receptor is the activated relay protein. This first protein kinase, going to break down ATP, breaks down ATP to activate number 2, which breaks down ATP to activate number 3. Well, look at this as a series of phosphorylations that are occurring to activate those enzymes, all the way down to a cellular response. So it's just a cascade, a series of phosphorylations that occur in this transduction pathway to get the cellular response. And that's as far as we go on this chapter. Okay. So that's the end of this lecture.